So the second part of this discussion of the metabolism, which we are going to take a look at an exemplary anabolic reaction occurring in the autotrophic organism, like a green plant. The process of the photosynthesis. The summary of photosynthesis is like the exact opposite of the reaction that what we have seen in the previous session, the oxidation of glucose. So here the starting material is the carbon dioxide, okay, and obviously you need water. Then it can miraculously, I would say, be converted into a more complex molecule, which is a glucose and an oxygen. Actually, this oxygen is a byproduct. So just like if we draw the arrow from the right to left direction, then exactly this is what exactly the cellular respiration or aerobic respiration is. So in that sense, it is a complete opposite reaction of. So if that respiration was an exemplary catabolic reaction, then this photosynthesis is an exemplary anabolic reactions. And obviously, as on any anabolic reaction, it takes an energy input. Then here, there is a reason why it's called a photosynthesis. The light energy, like a sunlight energy or any other light energies, are serving as an energy input here. That's exactly what's happening. And actually, surprisingly, is their entire reactions are strikingly like a reminiscent of those reactions that we have seen in the case of the cellular respiration. But then the starting part is how do we capture? It's not us. Unfortunately, we are not able to capture the light energy, and then that energy we can convert them in synthesizing something else, a more complex anabolic reaction. So we are not able to do this photosynthesis because we are heterotrophs, right? But just because we are an animal, that doesn't mean that we are not able to do this photosynthesis. Actually, some insects, it turns out that some insects are doing some photosynthesis. So it's whether you are able to do this photosynthesis or not is the presence or absence of this the light capturing pigment. Some of the molecule we call this a pigment. Some of the molecules are able to capture the light energy, and then it can transfer its light energy into to somebody else. Okay. In the case of a photosynthesis, that light capturing pigment, the major version of a light capturing pigment, its name is chlorophyll. Right. But this is not the only light capturing pigment, or another name for this light capturing pigment, especially in the case of photosynthesis, is photosynthetic pigment. So the pigment is the keyword. Chlorophyll is, there are two major forms of chlorophyll in the higher plants, chlorophyll A and chlorophyll B, but that's not important. But the chlorophylls are whatever is formed is that are the major photosynthetic pigments, and the reason why plant look green is this pigment. All those lights, there are some the specific uh, preferred wavelengths of light. In the case of this chlorophyll, they have some specific spectrum. The wavelengths of the lights are absorbed easily. But then some other portion of wavelengths of light, uh, if you think about this whole light, like the spectrum of light, when you look at the rainbow color, so this light, the visible light, actually consists of several different wavelengths as part of the light portion. So that like blue part and violet, blue, and the green and orange and red, like, and this order. So if you go toward the blue or violet part, the energy become higher. Then if you go toward the red part, then the energy become lower. The green is a kind of a little middle in the middle ground. So each different wavelength portion. This pigment can have some kind of a preferred range of this lights, the wavelengths, and in particular this chlorophyll molecule is very poor in absorbing the green portion of this wavelengths of light. That's why plant look so green, because it passes through or reflects particularly the green part of this light wavelengths. That's why it looks as it is, right? So it absorbed the red part, and it absorbed the blue part, and some specific wavelengths is there. On the other hand, another type of such light capturing or light absorbing or photosynthetic pigments, the group of such pigments are called the carotenoids. Actually, major carotenoids are so abundant in carrot, and the reason that's why the carrot has its own unique color of like a reddish or orange colors. All these carotenoids are also acting as an accessory supporting role in these light capturing activities. So in temperate region of the climate zone, in the fall, when those fall color of the leaves, the reason why yellow and then red beautiful fall colors appear is because at the time all those main light capturing pigment, which is the chlorophylls, are all destroyed to save the energy in preparation for upcoming winter. And then now those hidden minority of these accessory pigments of carotenoids appear now for the first time revealed, and that's why those leaves turn into the beautiful yellow or the red colors in the fall. In That's why only you can see those in only temperate climate region where these temperature changes over the year. Anyway, this is a modeling of when such pigments capture the 1 unit of light energy. So in other words, the point here is this photosynthetic pigment can capture the light in the form of a certain energy, right? So now, if you take a look at the structure of chloroplast, what's something that reminiscent is just like the case of the mitochondria. This chloroplast also consists of inner and outer membrane. Okay, so the outer membrane of a chloroplast over here, and also inner membrane of a chloroplast, the double membrane. In other words, just like the case of mitochondria, chloroplast also have this kind of intermembrane space, right? This. Hey, aren't we getting somewhere? Intermembrane space. So then you might wonder actually this intermembrane space may be able to have a little bit of a similar kind of occasion where you saw the hydrogen ion accumulation in this particular space, which ultimately drives the ATP synthesis. Kind of might be possible. Yes, that's exactly what's happening. So here in once we have figured out actually the basic mechanism of how the ATP is being synthesized in mitochondria through the electron transfer chain. Very similar thing happening in the chloroplast with exactly, I shouldn't say exactly, but basically the same mechanism of such hydrogen ion concentration build up in this intermembrane space. And actually, surprisingly, the same enzyme, the ATP synthase, through which the hydrogen ion can escape through, so then during which the ATP is synthesized from ADP, exactly the same enzyme is here in the inner membrane of the chloroplast, right? It's very interesting and surprising. So just like the case of the mitochondrial membrane, where inner membrane is highly folded, relatively speaking, but then that the folding of the inner membrane is much more extensive in the case of a chloroplast, so that's why you have see this some very complex building structure inside, some stacks of pancakes or stacks of coins or whatever you like try to make an analogy. These are collectively called this the stacking of this inner membrane is called the granum or grana. So this collectively to hold this inner membrane of chloroplast is called the thylakoid. In Korean it's called. So that's the inner membrane of a chloroplast, a specific terminology. And when that inner membrane is much more highly folded extensively, so that you have such a stack up structure, then you call that as a granum. Okay. But whatever they are, such specific individual terminologies are always something that is out of my interest in my class. So no one has to worry about memorizing such terms, but those are the terms that we have already decided to use for calling specific part of this chloroplast is the thing. So this the intermembrane space is what I said, just light energy. Okay? Somehow this light energy drives the accumulation of the hydrogen ion in this space between an outer and an inner membrane, and that accumulation build up this hydrogen ion concentration gradient to result in the ATP synthesis. So that's the one primary goal of this photosynthesis, because the energy provided by the light in the form of light can be utilized into the synthetic. So this is a first synthetic procedure. You are going to synthesize the ATP. Why? Because you need this ATP to drive the ultimate goal of this photosynthesis is to make the sugar, carbohydrate, the glucose, from nothing, which is carbon dioxide. That is one such prime example of an anabolic reaction, isn't it? So using that ATP, using this ATP, you can do this conversion of carbon dioxide into glucose. Is basically the bottom line of the photosynthesis. So this is once again my humble version of the chloroplast structure, wherein the whole entire chemical reaction procedure for what's happening in photosynthesis are all summarized here. So that's where you can look at this whole buildup of a hydrogen ion in this internal space between the outer and inner membrane, and that hydrogen ion buildup can also be extended into all this the space so this is actually intermembrane space too, all this green part. Okay? And to make this whole entire procedure a little bit more organized, what we can say is the entire photosynthetic procedure can be divided into two big stages. One is where actually the light energy is required, and then with that light energy you get to produce the ATP. This part is called the light reaction, yeah, because that's where the light is required. And similar to what's happening during the electron transport chain of the photosynthesis, what you see is this some of the membrane bound in the inner membrane bound protein complexes. What they do is like acting as a kind of an electron transport enzyme complex. Okay, very similar to those present in the inner mitochondrial membrane. So then you can figure it out. Okay, oh well, among these complexes, electrons will be transferred from one to the other. Yes, that's exactly what's happening. Look at this, right? So these electrons are like traveling between. Okay. And here what's happening is during this electron transport, also hydrogen ion accumulation also happen in this space, similar to what's happening in the mitochondria. And this hydrogen ion will try to escape through this enzyme complex. So now we all know that the name of this enzyme, the complex, this red guy, which is ATP synthase. And that's where this conversion of ATP from ATP is being made. So the ultimate result of this whole light reaction is the production of this ATP, right? And also another byproduct in most of the cases is the conversion of NADPH. NADPH is very similar to this NADH we saw in the mitochondria of the aerobic respiration, and its role is exactly the same. So it is some kind of an electron carrier cofactor. Okay, so from NADP to become NADPH, that means NADP is reduced now is into the NADPH. This was so these two are the products of the light reaction of the photosynthesis, right? One is perhaps the most important one is the ATP, and another product is NADPH, right? And both of them will be utilized in the second part of this whole photosynthesis, where actually the important thing, more important thing of sugar making, synthesis of sugar takes place. But also another actually thing happening over here is, although which is not exactly clearly illustrated over here, because not every plant and not every part of a plant there's something happening, but actually here another very important event actually to us, to all those heterotrophs like us, is the production of oxygen from water. Here, somehow this complex, using this light energy, they can split the water molecule, okay, into individual oxygen and hydrogen, and these hydrogens are actually can go into this little space. So this creation of an oxygen from water is actually very difficult chemical reaction, and we all know that the importance of this oxygen release as a result of the photosynthesis light reaction. Actually, this oxygen release, the production of oxygen is, the oxygen is a byproduct or the waste product, somehow drive this, the electron transport chain get going, and the victim of this electron, where are we going to take the electrons from? The water. Okay, so we are going to take the electron from you, but then that's where actually this difficulty of a chemical reaction. It is extremely difficult to strip oxygen in this water molecule, because the oxygen is not going to easily give out its oxygen in this occasion, but somehow plants figured out, or autotroph, photosynthetic autotrophs figured out how to do it, and thanks to that, actually, all these the oxygen dependent heterotrophs lives were possible. So we all know that how it happens. So that's basically what it is. So all this electron transport, very similar to the electron transport chain of aerobic respiration, is happening in the first part of the photosynthesis, we call the light reaction. And to drive such electron transport, this electron carriers is function of the activation of the electron carriers is done by this light energy, right? And just also another similarity is the location where it happens is the inner membrane of the chloroplast. And what I already have said, that is the name, the particular term calling the inner membrane of a chloroplast is the thylakoid is the one. So that's basically how we can summarize the entire light reactions. And once again, the acidification of internal, like here inside, just like the lumen of, so the space between outer and inner membrane, we call the thylakoid or lumen, internal space, intermembrane space, is highly acidified with all this hydrogen ion concentration build up, and all those build up hydrogen ion will try to escape. Okay. through this enzyme complex called ATP synthase, and that's how the ATP is being produced, right? Um, so that's the summary about light reaction. On the other hand, the second part of this uh, photosynthesis, we call those the dark reaction. It's because the second part of the photosynthesis actually does not require any direct light energy supply, and because of that, actually, to some plant, actually, this entire second part of the photosynthesis can be separated from, like, time-wise. The timing of this, the second part of photosynthesis, can be separated from the first part, which is the light reaction. The only thing it needs from the first part, the light reaction, is this ATP, the supply of ATP, and the NADPH. And surprisingly, this is another kind of a cycle. Although its name and slight nature of this chemical reaction, cyclic chemical reaction, is a little bit different. But in overall, it's kind of a backward reaction of the glycolysis, actually. So it's called the Calvin cycle. So anyway, this the second part, which is dark reaction, and the cyclic reaction's name is called the Calvin, the scientist who discovered the entire cycle in detail was the Calvin. His name was Calvin. So it's called the Calvin cycle. Is very also kind of similar to kind of similar to the cycle, the Krebs cycle or Tisei cycle in the aerobic respiration, and also the location is the internal space of this chloroplast, just like the other cycle reaction in the aerobic respiration happens in the matrix of the mitochondria. The only difference is here the internal space of a chloroplast is not called the matrix anymore, but there is another specific name of this internal space, which is stroma, is the terminology located indicating this whole internal space of the chloroplast. Once again, I don't really care whether you remember this term stroma or not. Okay? That's not really important. So eventually what's happening is some intermediate form of glucose will be made, and eventually it's converted into glucose, and then this starch is actually polymer of individual glucose molecule, right? So this starch is the storage form of this glucose molecule synthesized as a result of this photosynthesis, and starch is temporarily at least stored inside of the chloroplast, is what we can figure out this. So overall, the summary of this dark reaction is during the dark reaction of photosynthesis, carbon dioxide, what I mean by this, carbon dioxide, atmospheric carbon dioxide can be fixed, okay? So it's an apparent anabolic reaction, so you do need some of the energy input. That energy is coming from in the form of the ATP, which was made from the light reaction. And also this NADPH also can serve as another form of such an energy form. Some of you might wonder, they have in the cell can have already plenty of ATP because plant cells do also have mitochondria, and they can do this respiration when they are not involved when they are not actively engaged in the photosynthesis, they can respire during the dark to produce their own ATP. Why don't you use such ATP? Why do you really have to produce your own ATP to drive this glucose synthesis? First off, of course, it doesn't make really sense that in terms of the whole balance sheet, if you borrow some money from somewhere else, it's also kind of a debt. If you want to, uh, if you want to drive this reaction, the anabolic reaction, you better actually provide some energy from somewhere else to drive this. Is one reason, and another more important reason is the ATP is very difficult to move freely around in and out. So those ATP synthesized there might be a little bit difficult to have it easily transport into the chloroplast is another very good reason. Okay, uh, so that's the summary of this photosynthesis, and so is the whole overview of the two important metabolism as an example of the respiration and photosynthesis respectively. So the rest of this lecture, to finish up the whole discussion of a cell, is the discussion of the cytoskeleton. As I mentioned briefly, this cytoskeleton serves as some kind of an internal supporting role of a skeleton, just like our body skeleton. Individual cells do also have such kind of a supporting system, mechanical, plus alpha here. So it provides the function of such like our actual internal skeleton, bones, and the system, and plus some other very important functions are provided by this system of so-called cytoskeleton. And obviously, the function of the cytoskeleton is coming from proteins, some very specific, specialized protein, three different types of such proteins named. One is called microtubule, and the other one is called actin microfilament. So tubule here, one thing that we might want to actually intuitively distinguish between the tubule and the filament is like both tubule and filaments are long, pipe-like or thread-like long proteins. So in the cell, we can create many various variety of different shapes of protein, each of which can perform different function, right? Shape dictates the function. So that's what it is. But the difference between tubule and the filament is whether you have a hollow internal space with the larger diameter is a tubule, just like your whole the sewage and the water conductance pipelines have internal hollow spaces, and this inside the protein, the one major cytoskeleton protein, microtubule, has such a similar shape. So it has a hollow tube-like structure, long tube-like structure, is basically what this microtubule structure is about. On the other hand, the filament is like a thinner, thread-like long protein, is basically what we actually get to imagine when we try to imagine what the filament is. And so is this so-called actin microfilament. The reason why it is called actin is especially a lot of times this microfilament of the cytoskeleton we usually go by the actin, which is the particular type of protein from which this whole microfilament is made. So there is a tubule and a filament, and obviously some in terms of its size, there is an intermediate filament. So all these three different types of cytoskeleton proteins are all together making up of all the different variety of functions in the cell as a collectively called cytoskeleton. And so in the case of actin, so all these the cytoskeleton proteins are long big polymers, and we have already seen in many different other occasions that when it comes to building up a polymer in the cell, you get to start small subunit, small monomeric units, so that in the case of starch, when you need to build up starch, then you build up monomeric form of glucose and then combine, assemble all together to make a long thread-like structure of starch. When you make a DNA strand, then you collect individual nucleotide and then assemble them so that to make a long thread-like DNA. So it's when you want to build up protein, and then you gather 20 different kinds of amino acids and then assemble in some specific order, and then that's how you make up a protein. And in this case, the cytoskeleton protein is such a huge, much larger structure. So each protein, individual monomeric form of protein itself is actually already polymer. However, using that individual polymer, like here in the case of actin microfilament, each ball-like structure is an actin subunit or actin monomer to build up long filament like just such a structure is how you generate this actin microfilament. Okay, and similarly, in the case of this intermediate filament, you have some of those monomeric form to combine together have such an intermediate filament. By the way, actually, that the progeria the defect, genetic defect, resulting in the premature aging syndrome due to the defect in lamine. The protein, the particular protein underlying the inner surface of nuclear envelope that we have seen in the previous lecture, is actually one of the such example of this intermediate filament, the cytoskeleton function. So hopefully you begin to recognize the importance of these cytoskeleton functions in the cell. Okay. And finally, this microtubule, which is the biggest in terms of its size and the structure. Also, to build this whole hollow, big tube-like structure, you start with this such smaller subunit form of a protein. Its name is a tubulin. The name is not important at this point. Is the thing. So here the point is there are three different types of such cytoskeleton protein, each of which is assembled from monomeric. Each specific monomeric form of the protein is the whole summary of this whole point here. Now, what they do is more important. So the function of the cytoskeleton is first off, it's the one of the most important function obviously is the shape maintaining the shape of the cell especially becomes obviously important in the case of the animal cell where there isn't any rigid outside of a supporting structure like cell wall. Animal cells don't have any rigid cell wall outside of the cell membrane. So, for example, there are many different forms of animal cell shape. Like, for example, some animal cell shape like this, like face of the Simpson, uh, having at the top it's like a finger like a projection. At the bottom, relatively flat bottom. And how do you generate this particular finger-like projection is one is all about inside you have some specific lining of cytoskeletal protein. Some other animal cell, for example, especially in neuron, your nerve cell have this particular shape. Now, how do you have this particular shape of this over here and over there? It's all about how do you make a specific overlay inside of this cell with particular form of particular arrangement of cytoskeleton protein. So that's where this maintenance of a shape function comes and become important. And also another lot of times actually ignored important function is in the cell, the organelles. Some specific organelles are held in check, held at a very specific position in the cell. Otherwise, these specific organelles will, like so to speak, will float randomly here and there. And as we have seen, like in the transport system of, for example, like exocytosis or endocytosis, those destination, those organelles serve as some of the intermediate destination of this whole procedure. And if they move around, it's not really efficient and good. So some long stick like this cytoskeleton protein, like kind of make specific particular organelle in position is another important function of this. However, more impressive and important function coming from the cytoskeleton would be this, maybe personally. It provides also the trap for this transport vesicles movement. Like as we have seen in the case of the exocytosis and endocytosis as well, all these different cargo delivery materials are shipped in the form of these transport vesicles, right? And these transport vesicles are delivered from one place to the other, for example, like from rough endoplasmic reticulum to the Golgi. And how do you send this transport vesicle from here to there? It's like a boat on the water, so you just push this whole transport vesicle from here to the other side, and then it will just float over there and then eventually reach the other shore. Is that maybe you might have actually one like imagined that is the way, but no, it is not actually. It follows, each transport vesicle follows some specific track. You can imagine there is a kind of a railroad tracks or maybe some highway track or some local road, whatever, to your preference. But there is some specific paths are there, all in between among different part of the organelles and the location in the cell, and that path or tracks are provided by this cytoskeleton protein is the key. And then how those transport vesicles move along the tracks. Here comes this the motor proteins. On specific type of cytoskeleton protein, there are some specialized only working on that particular type of cytoskeleton protein. There are some specific forms or types of motor proteins exist, and those motor proteins grab each individual the transport vesicle and then move step by step. It works actually, and the energy of this movement obviously come from ATP hydrolysis. The energy ATP. So there is another very good area or where this energy ATP in the form of ATP is required. So that's is another very important function of this cytoskeleton protein. So this by now you can see this is an example of this one particular type of cell, which is an epithelial cell of animal. This epithelial cell is the one covering the lining of external our surface, the of a body, and also internal the organs body. So like our intestine, the surface of our intestine is covered by this epithelial type of epithelial cell, and that's why in epithelial cell you do have this, at least one area have this highly extended surface area to increase, to have a larger surface area, so that they can have a larger area of this material exchange. It would be much more beneficial, especially if you consider this in like your lining of your intestine. Then you can absorb more nutrients with the same volume of the cell area, isn't it? So anyway, inside, if you look at, hopefully you will be able to recognize and distinguish three different types of the cytoskeleton protein in different colors. So here in sky blue, proteins represent the microtubules. Along with that, like this is motor protein here that I mentioned. This motor protein is the one actually carry each individual transport vesicles along this microtubule. And also another different types of motor protein do exist in the actin microfilament, where actin microfilament has formed its organization in a different area of the cell, and some of the intermediate filaments to provide the strictly supporting structural role, as you have seen over here, like that. Okay. So this is the overall the function of the cytoskeleton protein that I have just mentioned: maintenance of the shape and organelle check-in position. You maintain the specific location of the organelle and the railroad tracks, the paths for movement of all these different transport vesicles right there. But that's not it. And that same function also can be seen in this example of the nerve cell, the neuron cell. The reason why neuron cell has some very specific, interesting looking shape is due to the arrangement of specific cytoskeleton proteins. The same color coding scheme was also employed over here, so that we can see here mainly microtubule and intermediate filament can be seen over here, whereas at the terminal end of the nerve cell you can basically see lots of these actin filaments can be seen like this. But additionally, the cytoskeleton also provide a function of cell division function. When cells try to divide, actually, this role of a cytoskeleton become also essential. Okay. So during the cell division, that's where another occasion where this cytoskeleton proteins are needed. And also when individual cell try to move, so motility, movement, okay? And also as an kind of organized tissues like muscle, when this muscle provides some of mobility through this muscle contraction, that's where actually particular form of the cytoskeleton proteins uh, provide the needed function. So let's take a look at first the cell division function. Here, this is a kind of a diagram of an animal cell is about to divide into two. Okay. So when you try to divide, the first mission is you have to distribute your chromosome equally into two other areas. So this area and this area will become eventually two different individual separated entity at the end of this cell division, right? So that you have to transfer uh, this chromosome, send to this direction, whereas this chromosome you have to send the other opposite direction. How? How can you do? Like you attach some of a string on a puppet and then pull them down is how you do it. So each these chromosomes, once again, same color coding scheme. So this, all this, the sky blue thread-like structure must be microtubule. Yes, microtubules are actually specifically formed. Here, these microtubules are something different from those microtubules used for the railroad track in the cell. This is some new version of the microtubule. We call this a spindle fiber. But whatever you call it, it is the same microtubule that you have seen in the case of such a cytoskeleton of that provide the function of the tracks for the transport vesicle movement. So this spindle fiber is the one actually each of this spindle fiber attach hook to the individual chromosome and then pull each individual chromosome to the desired location is how you separate and correctly distribute this chromosome. By the way, the Korean name for this spindle fiber is this. Right. On the other hand, if you want to separate these two animals, the animals into two, then you have to cut or sever at the middle point of this animal cell. But the way you do accomplish is squeeze this animal, because animal cell is only like made up of soft cell membrane. So you squeeze a certain specific area of the surface of the animal cell to contract to become like you just tighten up your belt, waist belt, just so hard and keep on doing that, you may be able to actually cut your the waist so that your upper body and the lower body become two separate identity. Is it too cruel example? I don't know. So that's how you do, and where this actin filament is required. Actin filament is very specialized in function of contraction. So then it somehow specifically squeeze, in other words, contract, so that make this thing is very narrow, narrower, and then cut. Finally, eventually cut it. That's how you accomplish this entire cell division. Speaking of a movement, amoeba. Here in amoeba, some surface has a kind of flexible projection and then retraction, and that's how you, as an amoeba, how that's how you capture your food, your prey as a food, and then retract so that internalize through sort of the endocytosis or phagocytosis that we have seen in the previous lecture. And this extending and then retracting this projection quickly is actually the work of the rapid synthesis of actin microfilament, and then rapid degradation of such an actin, and then that's how you extend your certain specific surface and then grab and then retract. It's such kind of a movement. It's amazing how quickly can you accomplish such like a synthesis and then the degradation of actin filament inside, and you accomplish your mission somehow through that. But that's what you do exactly. And our own white blood cell, similar to this amoeba, some of the defense white blood cell do also this type of a thing, and that's how they do this phagocytosis to get rid of any invading pathogens. And also, this whole lot of hairy structure is also is a microtubule. It's called specific type of microtubule called the cilia. Okay? And not only the cilia have so many numerous cilia cover of the surface of this particular microorganism, and all together overall by creating some the waving movement, it provides some of the motility movement function. But also in our internal like the throat, esophagus, and the internal the respiratory tract leading to up to our the lung is also covered, the surface is covered with many cilia protein, and they also do generate this wave-like movement to create a constant flow of liquid inside, and that's extremely important in maintenance of our overall health against pathogen infection. That particular example we will get to see in the following chapter later on. But so anyway, bottom line is microtubule also provides such movement function, and also individual sperm cells movement is also provided by this long microtubule. In this case, particular type of microtubule's name is called not the cilia, but it's called the flagella. But once again, names are not important. Microtubule and microfilament actins are the ones that are acting in generating this type of a movement. And finally, look at this. When it comes to this muscle contraction, the actin filaments are all organized in the very like one very thick unit of contraction, and that's basically what your muscle fiber is organized and how it looks. But the difference here is, unlike in the case of actin and its own the motor protein, here the myosin is the name of special motor protein working only on the actin filament. but their roles are somehow they are the kind of a reverse, so that all these motor proteins are combined, many numerous motor proteins are combined and held together, um, and there is a thinner actin filament, and then these motor proteins are all together provide some of the contracting motion using the energy, the ATP hydrolysis, and that's how you pull this actin filament. So that the contraction means you are pulling this actin filament into to become more shorter, like so that this actin can be slided into this area. So the overall the individual unit length was this long, and then after the contraction this individual unit became this much shorter, is how you achieve this muscle contraction. After all, I mean, the details like this that I just like babbled like this is not really important at all in at this level, and at this moment. But to provide some version of explanation about how this muscle contraction is achieved through this actin, that's why I kind of went into a little bit more details. If this were on a general biology, like general biology 2 course, then I would have actually gone into very specific details about how this muscle contraction is being accomplished. But for now, yeah, we don't really need such unnecessary details. Okay, with that, so we can finish the entire chapter of this chapter 4. And although I think the time wise, we have a little bit of time left, so that we can jump into the new chapter of this DNA metabolism and gene expression control. But I will just refrain myself over here and stop over here for this week's lecture. Only wishing you to have a nice weekend, and see you next week.